Welcome to this lecture on the biochemistry of metabolic processes. Today, we are going to explore the fascinating world inside our cells to understand exactly where and how energy is created. We will look at the specific sites of ATP production during oxidative phosphorylation. This is the ultimate key to understanding how the food we eat is transformed into the life-sustaining energy that powers every single thing we do. Let us dive in. Now that we know what we are studying, let us start with a simple analogy. Imagine a massive hydroelectric dam holding back millions of gallons of water in a high reservoir. The water naturally wants to flow down to the lower river, but it is completely blocked by the thick concrete wall of the dam. When the engineers open a small, specialized gate, the water rushes through with immense force, spinning a giant mechanical turbine that generates electricity for entire cities. In your body, your cells do the exact same thing to create energy. Building on that dam analogy, let us look at what is actually happening inside your cells. Instead of water, your cells use tiny positively charged particles called protons. The dam in this scenario is a tightly sealed membrane located inside your mitochondria. Your cells actively pump these protons across the membrane, trapping them on one side. Because they are positively charged and highly concentrated, they desperately want to flow back across, creating a massive chemical and electrical gradient. This gradient is a huge reservoir of potential energy, just waiting to be unleashed. So, how do these protons get pumped across the membrane in the first place? When biochemists talk about the sites of ATP production, they are referring to specific protein complexes embedded within this inner mitochondrial membrane. Historically, scientists identified 3 distinct sites where the energy from electron transfer was captured: Complex 1, Complex 3, and Complex 4. We now understand that these 3 complexes are actually powerful proton pumps. As electrons from our food pass through them, they use that energy to push protons out of the mitochondrial matrix, building up that massive gradient we just discussed. With the proton gradient fully established, we finally reach the actual physical site of ATP synthesis. This is a separate, magnificent structure called Complex 5, or ATP synthase. Think of this enzyme as the brilliant molecular turbine in our dam analogy. It acts as the only open channel that allows the trapped protons to flow back into the matrix. As the protons rush through, ATP synthase physically spins. It uses that kinetic energy to forcefully attach a phosphate group to ADP, thereby creating ATP, the primary energy currency of your body. You might be wondering, why does it matter exactly where and how ATP is produced in the mitochondria? Well, if our bodies relied solely on the simple breakdown of glucose in the cytoplasm without mitochondria, we would only get a measly 2 molecules of ATP per glucose molecule. That is barely enough to keep a simple single-celled organism alive. By utilizing these specific pumping sites and the ATP synthase turbine, our cells can extract up to 32 molecules of ATP from that same single glucose molecule. This massive energy boost is what allows humans to have large, complex brains, beating hearts, and the ability to run marathons. Because this massive energy boost is so critical, these specific sites of ATP production are also our biggest vulnerability. Furthermore, many deadly poisons, like cyanide or carbon monoxide, kill humans specifically by blocking these exact ATP production sites. When these complexes are blocked, protons can no longer be pumped. The gradient collapses, the ATP synthase turbine stops spinning, and the body's power grid shuts down instantly and fatally. This perfectly illustrates just how essential these specific mitochondrial complexes are to our survival. Now that we understand the biological machinery, we need to know the exact numbers of protons involved before we can calculate efficiency. Laboratory experiments have shown us that when the electron carrier NADH donates its electrons, they travel through complex 1, 3, and 4, pumping a total of 10 protons across the membrane. However, another carrier called FADH2 enters the chain later, skipping complex 1 entirely, which results in only 6 protons being pumped. Finally, we know that the ATP synthase turbine requires exactly 4 protons to flow back through it to rotate fully and synthesize one single molecule of ATP. Keep these numbers in mind. Now that we know the exact proton numbers, let us look at how we mathematically measure the overall efficiency. To quantify exactly how much ATP is produced at these sites, biochemists use a mathematical value called the phosphate-to-oxygen ratio, represented by the letter R. This ratio measures the overall efficiency of oxidative phosphorylation. In this equation, N sub ATP represents the number of moles of ATP synthesized by the ATP synthase turbine. N sub O represents the number of moles of oxygen atoms that are reduced to form water at complex 4. Oxygen is the final electron acceptor, so measuring it tells us exactly how many electrons have flowed through the pumping sites. If N sub ATP increases while oxygen consumption stays the same, the ratio R increases, meaning the mitochondria are highly efficient. Let us apply these numbers to calculate the theoretical ATP yield when one molecule of NADH donates its electrons. We are given that NADH pumps 10 protons, and 4 protons are needed per ATP. First, we find the total ATP produced, N sub ATP, by dividing the 10 protons pumped by the 4 protons required. This gives us 2.5 molecules of ATP. Since one molecule of NADH passes its electrons to exactly one oxygen atom to make water, our N sub O value is simply 1. We substitute 2.5 and 1 into our efficiency equation. The final answer is a ratio of 2.5. Physically, this means every NADH molecule successfully manufactures 2.5 molecules of ATP for cellular work. Now, let us look at a different electron carrier, FADH2, to see how the entry site affects the yield. Because FADH2 skips complex 1, it only activates the proton pumps at complex 3 and complex 4. As a result, it only causes 6 protons to be pumped across the membrane. Our strategy is identical to the previous example. We divide the 6 protons pumped by the 4 protons required per ATP, which gives us an N sub ATP of 1.5. Dividing this by one oxygen atom gives a final efficiency ratio of 1.5. This physically means that FADH2 is less efficient at driving ATP production than NADH, yielding only 1.5 molecules of ATP. The specific entry sites dictate the final energy output. Having calculated the exact energy yields, let us visualize this entire process together. Here you are looking at a detailed schematic of the inner mitochondrial membrane. Notice the lipid bilayer separating the matrix below from the intermembrane space above. On the left, you can see complexes 1, 3, and 4 acting as proton pumps. The upward arrows clearly show protons being pushed into the intermembrane space, building that high concentration gradient. Then, on the far right, look at complex 5, the ATP synthase. You can see the protons flowing back down through it, driving the physical rotation that forces ADP and phosphate together to manufacture ATP. This diagram beautifully captures the cellular hydroelectric dam in action. We have covered a lot of ground today, so let us summarize the most important ideas. First, remember that the sites of ATP production are intimately linked to the electron transport chain. Complexes 1, 3, and 4 act as the crucial pumping sites that build a massive proton gradient. Second, complex 5, or ATP synthase, is the actual manufacturing site where this gradient is converted into ATP. Finally, by understanding the math behind the phosphate to oxygen ratio, we can clearly see how the number of protons pumped directly dictates the amount of life-sustaining energy our cells can produce. You now have a complete understanding of how your body extracts energy at the molecular level. Great work.